So thank you for the nice introduction and good afternoon. So I will be presenting on the large-scale virtual brain modeling for personalized optimization of deep brain stimulation. So over the past 30 years, brain stimulation has attempted in various modalities. In various modalities to treatment and neurorehabilitations for various neurological and psychiatric disorders. So many studies have shown the promising results and demonstrated its effectiveness, but to date, the clinical outcomes still have varied across studies and individuals. So many neuroimaging and computational studies are actively being conducted to improve their therapeutic efficacy. So these advanced techniques include refining the exact target area or the white matter tracts by considering the anatomical structures of individual brain, or optimizing the stimulation site or parameters through the computational simulations to induce the highest current to the target brain area. But the brain stimulation not only produces the local effect in the stimulated areas, but also induces the network effect through interregional interactions mediated by brain connectome. So to investigate this network effect encompassing structural and functional properties as well as dynamic changes due to stimulation, we used the virtual brain modeling approach. So just briefly to introduce the workflow of the traditional personalized virtual brain modeling. First, we need structural brain reconstruction based on individual MRI data. So this movie was constructed by my brain images, inspired by the virtual brain and human brain project workshop. So from the T1 weighted images, the cortical and subcortical structures are reconstructed. And from the diffusion weighted images co-registered with the T1 images, nerve fiber tracts are reconstructed through the tractography algorithm. And then based on brain parcellations by specific brain atlas, the number and average length of fiber tracts connecting different brain areas are calculated, and through this, the individual brain connectivity is derived. So next, we need a neural mass model to describe the dynamics of each brain region. So it is a mathematical model to reproduce neuronal populations activities. So these neural mass models are placed into each brain region and linked by the reconstructed individual connectivity by considering connection strength and transmission delays. And then because the intrinsic characteristics of each brain region may be different, so regional specificity can be reflected as a parameter of neural mass model of each brain region. At this point, data-driven results from the functional recordings or clinical hypothesis can be applied. And furthermore, machine learning techniques can be used to estimate the parameter values of each brain area. Then finally, we have the personalized virtual brain model. So using such a model, we can simulate the individual resting state network or the stimulation-induced responsive networks by applying the stimulus to the into the specific brain area. Then the simulated signals can be projected into sensor space, such as EEG, MEG, fMRI, through the board modeling, for the direct comparison with empirical data and further analysis. So from now on, I will briefly introduce a proof of concept study where we applied the personalized virtual brain approach to the deep brain stimulation for treatment-resistant depression. So major depressive disorder is known as a brain network disorder involving structural and functional abnormalities, and 30% of patients are classified as having the treatment-resistant depression, TRD. So for those patients, DBS has been applied over the 20 years with the multiple target areas. The most commonly used target area is the subcallosal cingulate cortex. But so far, the results have varied across studies with an average response rate of 40 to 60%. So many neuroimaging studies are being conducted to improve its efficacy in personalized strategies. In particular, Helen Mayberg group revealed that responders to DBS shared specific white matter tracts from the stimulation site. And through EEG recording, they also demonstrated that when those fiber tracts are activated, then specific cortical evoked potential patterns can be generated. A more recent paper has shown that distinct cortical EP patterns are observed depending on selectively stimulated fiber tracts. For example, selectively stimulated cingulum bundle or forceps minor, something like that. So these studies suggest the potential use of the cortical EP patterns as an indicator of the target white matter tract engagement. So inspired by this existing study, we started the modeling study. So using this personalized virtual brain model, we can identify the fiber tracts affected by stimulation and then simulate the responsive network induced by selective activation of these fiber tracts. So we thought that if the target EP associated with the DBS responsiveness is well established, then we can systematically change the stimulation site and observe the resulting EP patterns and suggest the optimal stimulation condition that reproduce the EP patterns most similar to the target EP. So this was the concept of our study. So as a proof of concept study, we simulated these cortical EP patterns according to the stimulation site. So here we used data from three patients who are already receiving DBS treatment. So we used the implanted electrode contact positions as the different stimulation site. But to achieve this, we extended the traditional virtual brain modeling framework by combining two factors, high resolution and explicit fiber tract modeling. So the high resolution model was constructed by placing neural mass models into the vertices of brain surface mesh, regardless of the specific brain atlas. And through the explicit modeling of fiber tracts, considering location and their geometries, each fiber tract could be active and selectively stimulated. So here we used a simple generic 2D oscillator model as a neural mass model. And by calculating electric field from the stimulation side, we selected the fiber tracts to be activated and triggered non-network activities by applying the instantaneous input to the neural mass model at the both ends of these selected fiber tracts. And then simulated signals from each neural mass model were projected into patient-specific sensor space. So these are simulation examples in which different stimulation point, like L3 or R1, was used as the stimulation site, respectively. So due to the activation of fiber tracts, the locally initiated activities are propagated to other brain regions through the individual connectivity, generating a specific spatiotemporal response pattern according to the stimulation locations. So we performed this simulation for 8 different electrode contacts and identified distinct EP patterns. And we explored the EP patterns by sweeping 2 modal parameters, global coupling for the connectivity and stimulus input gain, and calculated the similarity value between simulated EP and empirical EP signals based on Pierce correlation. So when using the optimal parameter values for individual patient, the model showed differential accuracy across the stimulation positions. So as you can see, the similarity value showed higher values around 0.8 at some locations, but showed lower values less than 0.2 or even minus values at some other locations. So this was our study's limitation. However, one thing I should note is that the locations showing lower similarity values corresponded to the locations showing very small response signals in the empirical data, and those were electrode contact that had been used for DBS. Long-term DBS is generally known to suppress neural activities around the stimulation site and induce the neuroplasticity accordingly. But in this study, we just used diffusion-weighted images at least 3 years before the EP experiment, meaning that we used the connectivity information that did not any potential neuroplasticity that may be induced by the long-term DBS. So we thought this might contribute to our limitation. So despite many limitations, this study has significance in that it provides a theoretical framework to describe the link between fiber tract activation and stimulus evoked potential, which are crucial factors for DBS treatment, at least in TRD patients. So now we are currently working on resting state simulations to investigate the modulation effect induced by the long-term therapeutic stimulation. So thank you for your attention. And I would like to express my appreciation to our colleagues in Theoretical Neuroscience Group in INS for many discussions on methodology, and also research groups in University of Calgary for providing their valuable clinical data. So thank you again.